Okay. Good evening. Welcome to the IOPPN and the Wolfson Lecture Theatre. This is a very special event in the calendar of the IOPPN, the Paul Janssen Lecture, named after the founder of Janssen Pharmaceutical Company and the inventor of haloperidol. And every year we have a very distinguished VIP superstar academic speaking at this event. And this year it's my pleasure to introduce Professor Jonathan Mill. He's from the University of Exeter Medical School. I know a lot of you already know him because of his time when he worked here at the IOP. He is the youngest ever speaker we've had at this event. Usually it's an older generation of academic, but because he's so brilliant, he's already achieved more than some of his more senior peers, so that's why he's here today. And his expertise is really in the field of epigenetics. So Jonathan started his life as an undergraduate at Oxford. He then did a PhD in psychiatric genetics here at the IOP. Then he went to Toronto to do a postdoc for 3 years, and that's really where he really got into epigenetics. And then he came back to the IOP and set up the psychiatric epigenetics unit, which he still heads, even though he's since moved to a chair in Exeter in 2012. So his research is that he's really pioneered the use of epigenetics in mental health, and his research really focuses on how epigenetics can mediate gene-environment interactions in mental health disorders. So it's relevant to really everything that goes on at the IOPPN. So I think his talk will be relevant to potentially everything and everyone. So without further ado, Jonathan, welcome. The floor is yours.
Thank you very much for the intro. Can everyone hear me at the back? Just checking. Yes. Brilliant. So it's a real honour to be here giving this lecture, having spent time in this lecture theatre when it was a bit scruffier looking as a student, and then when I was a lecturer. So I'm really glad to be here talking to you about my research today. And I just want to start with probably the most important slides. This is my group in Exeter. And the focus of our work is really on understanding both the causes and consequences of molecular variation in the brain and the role that plays in a range of neuropsychiatric and neurodevelopmental and neurodegenerative disorders. And really a kind of really good starting point for this talk is really just to highlight the amazing advances that you're probably all aware of in understanding the genetic basis of mental health phenotypes. So this just shows really the progress that has been made by many people working here and elsewhere within the Psychiatric Genomics Consortium. Really highlights the power of collaboration in science, and by this area, people putting together large sample sets and starting to really identify robust regions of the genome with harboring genetic variants associated with disease. And I think the most recent meta-analysis is showing that there's over 250 regions of the genome harboring robust genetic variants for schizophrenia. And actually, if you told me that when I was a student 15 years ago here, I think that would have been almost unbelievable. But really, the kind of key challenge is now to move beyond this and to really improve understanding of the actual biological effects of these genetic risk variants. That's kind of one of the themes that I'm going to talk about today. And of course, we, for not just schizophrenia, for complex disease in general, we really are now starting to get a good handle on the polygenetic risk factors that mediate disease. And obviously epidemiological work, again, much of the kind of leading work in that field in the context of mental health is done here, is really starting to highlight some of the robust environmental factors that lead to risk of disease. But how these kind of things come together to bring about changes in the way that genes function in specific cells in the brain at specific points in our life course, perhaps during aging and development, is still really much a black box. And it's really those molecular processes that our work is trying to understand. And actually, the kind of first realization that something else is going on in the context of schizophrenia was really done by two people that probably people here know very well, Irv Gottesman and James Shields. And they were the first to really mention the term epigenetics in the context of schizophrenia. And their kind of understanding, or the molecular understanding of what epigenetics is, wasn't really very well developed at that stage. But they really kind of, I think, had some really key statements that really address some of the things that we're now trying to address. So they mention that both genes and environment are essential for the development of any human characteristic, and that this puzzle is what they call epigenetic, and that the task ahead is really to discover how genes and environment interact to produce schizophrenia. And they also had, I think, a really pertinent point here, and this is something that I'm going to touch on a lot in the later stages of the talk, and that's really to highlight how difficult it is to actually elucidate these mechanisms. So they say, of course, it's much easier to talk about such epigenetic interactions than to design experiments that will elucidate them. And I think that'll become clear in some of the examples I give. And this is a figure from the conclusion of their book, and it again touches on some of the things that we're very interested in looking at. So they show how people with maybe different genetic liabilities for schizophrenia, maybe high genetic burden or low genetic burden, through different mechanisms over the course of development can end up maybe with different levels of the disease. And one of the things that they're particularly interested in, and again I'll touch on this later, is the fact that you can have two individuals with the same exact genome, genetically identical monozygotic twins, who can end up discordant for a disease. So across the kind of spectrum of complex disease, we know that things other than DNA sequence variation are likely to be important, and that comes from the observation that identical twins are often discordant for disease or for clinical symptoms or other features of a disease. We know that many complex diseases, particularly various mental health phenotypes, show clear gender differences, both in prevalence and symptoms. We know these diseases show a fluctuating disease course, so they come on and off during different points of the life course. We often see peaks of diseases at certain times in life, perhaps following periods of hormonal changes. And of course, we know that the environment's important, but it appears that the environment may be particularly important at very vulnerable, critical periods in development. And so all these things suggest that mechanisms other than DNA sequence variation may be important. So this is how genetics, GWAS studies see the world. This is a kind of one-dimensional sequence of A's, C's, G's, and T's. And obviously what disease studies are trying to do is to look for differences in this genetic sequence and see if they're more frequent in groups of individuals who have a disease compared to individuals who don't have a disease. And we know that this type of genetic variation is really what drives evolution. So we can now sequence whole genomes, and we can pinpoint the exact genetic differences that differentiate us from, say, our closest evolutionary neighbors. But actually, if you take a brain from a human and a brain from a chimpanzee, they're both clearly very much the same organ. But any of us here is made up of maybe 200 different cell types. Each of these comes from exactly the same genetic code, so every cell in your body has the same DNA sequence. But if you look at a neuron, the cell types that maybe we're interested in, under a microscope and compare it to a blood cell or a fat cell, you'll see that they look very, very different. And this is a result of the different set of genes that are activated in any of those different cell types. And of course, it's even more complex than that. So if you take the central nervous system, we now know that there's a kind of huge amount of diversity of different cell types within the brain, and new technologies such as single-cell RNA sequencing approaches are starting to allow us to actually characterize these many different cell types within the human central nervous system at the gene expression level. So you only really have to kind of take a step back and actually look at how our genetic material, our DNA, is stored and packaged to realize that it's much more kind of complex than a linear sequence of A's, C's, G's, and T's. So this is a really beautiful picture of a human mitotic chromosome, and you can see the ornate way that the DNA is stored and packaged within the nucleus. So DNA is coiled around these core histone proteins, which in turn are folded together to form the chromatin and the actual chromosome structure. And it's this structure, or the way that the DNA is packaged and stored, that really controls how accessible the DNA sequence is to the factors in the cell that bind to the DNA and actually drive gene expression. And it's actually this at its kind of most basic level that we're studying when we talk about looking at epigenetic modifications to DNA. So just a bit of history. The term itself was really first coined by a British biologist. This is Conrad Waddington, and he used this concept of what he called an epigenetic landscape and a marble rolling through this landscape to really describe how cell fates are established during development. So going from a state where they're totipotent, so they can become all the different cell types that make up any of us, to that point where they become fully differentiated into the specific cell types that make up an organism. And there's many analogies you could take, but this is probably a train junction that's familiar to many of you. This is Clapham Junction in South London. Apparently the most busy rail network or junction in Europe. But you could imagine how a train going down these tracks, if it was set off by the wrong signal being switched on, or going to the wrong platform or the wrong direction, could end up down a very different place in the country. So this is a kind of analogy really of how these cell fates are established during development. Another way to think about your epigenome is to think of your DNA sequence as a piece of sheet music, the kind of phenotype, the way that those genes are ultimately expressed as an orchestral performance. And the epigenome really is the way in which that piece of music is interpreted to go from genotype to phenotype. And you may ask, why is this important, and why might this be important for disease? Well, imagine two identical stretches of DNA. On the left, the epigenetic signature around a gene is such that there's lots and lots of gene expression, lots and lots of protein being produced. On the right, a very different epigenetic scenario, which means to there being no gene expression and no protein being produced. So the kind of take-home message here is it's your DNA sequence that determines the specific mRNA molecules that are actually made or expressed. But it's the epigenetic regulation of gene expression that determines how much or when and where in the cell or the body that mRNA is made. And so the kind of follow-on to that is that even genes that don't have any disease-causing mutation or variant within them could still be pathogenic if they're expressed at the wrong time or at the wrong amount or in the wrong place. So in the context of understanding kind of human health and disease, most of the focus has been on a modification to DNA, DNA methylation. My pointer isn't working. I don't know if there's another one, but I'll use it. Essentially, this is the addition of a methyl group here to the fifth carbon position of cytosines. That's one of the four bases that make up our genetic code. And the actions of this are really first are twofold. One is that they block the binding of these transcription factors that bind to the DNA and drive gene expression. And they also attract certain proteins that, again, alter that structural conformation of the DNA that I showed you earlier. And we now know, actually, there are many more modifications to DNA. In particular, in the central nervous system, we're very interested in what we call DNA hydroxy methylcytosine. This is a modification that only seems to be present at high levels in the central nervous system, and particularly in neurons of specific regions of the brain. It appears to be a very stable modification in the brain, suggesting that it possibly plays a role in neurobiological function. And in fact, there's a kind of whole suite of epigenetic modifications that don't so much act to switch on or off genes, as textbooks may say. They actually really act to almost fine-tune genomic function over the course of development and in different cell type specific states. And there are a lot of efforts that have kind of followed on from the Human Genome Project that acted to map the genetic code that make up humans, that are now moving on to characterize these epigenetic modifications across the genome, across all the cells and different tissues that make up any of us. So this is a figure from the RoMap Epigenomics consortium paper. We were involved in mapping some of the patterns across different regions of the brain. And what you'll see here is for one kind of region on the genome, each of these layers here is a different type of epigenetic modification. And the details here aren't important, but it's that unique combination of epigenetic marks sitting above the DNA sequence that determines the specific gene sets that are activated developmentally in those cell types. So you can really think of this as generating an atlas of gene expression across all the different cells and tissues that make up a human. And the key take-home messages from this work are that different cell types are characterized by very distinct epigenetics, and these mirror the developmental relationships between cell types, tissues, and lineages. And they're the common developmental origin as the primary determinant of these global epigenetic patterns. And one of the limitations of the work that's been done in this context so far is that there's been very little focus on the cells and tissues that we're particularly interested in neuroscience and mental health. And so the PsycEncode consortium was formed to really start to characterize patterns of gene expression and gene regulation across the different cells and tissues that are present in the central nervous system, to look at these across development and then relate these patterns of normal gene regulation and expression to some of the diseases that we might be interested in, including schizophrenia and autism. So one of the things that makes the kind of epigenome or gene regulation so appealing to understanding the development of disease is that while your DNA sequence is very stable, so as I said, every cell in your body will have an identical DNA sequence from the point of conception onwards, other than the kind of few random somatic mutations that occur, patterns of gene regulation are much more dynamic. And over the course of an individual's life, they can be influenced not only by development, but also by factors in the environment, by immune responses, stochastic or random changes, and even DNA sequence variation itself. So it's kind of one potential route by which two genetically identical individuals could end up very different in the context of gene regulation and ultimately impacts upon health and disease. So I mentioned the environment, and for many people, this is really what the interest in epigenetics is about. This is what they're really interested in understanding. And there's now kind of growing evidence that a whole range of factors in our environments can directly influence gene expression and gene regulation via changes to the epigenome. I've put question marks up there because this is still really a very kind of nascent area of research. But one of the kind of key take-home messages from this work is that the developing individual appears to be particularly vulnerable to these external environmental exposures at very critical periods during their life. And these kind of map very nicely onto what we know about critical periods of sensitivity for complex disease. And a really important period appears to be very early in pre and perinatal development. And if you think about it, this makes sense. It's the time that there's lots of cell division taking place, lots of DNA replication, the time that those standard epigenetic marks that define all our tissues and cell types are actually being laid down and established. And because these modifications can actually be very stable once they're established, this provides a very nice mechanism by which exposures very early on in life may manifest in the context of gene expression differences or functional genomic differences across the life course. And this has been touted as kind of one mechanism underlying some of the developmental origins of health and disease. And of course, in a disease like schizophrenia, we know that there are these very critical points across the life course when very specific environmental factors seem to be particularly important in mediating risk of developing disease. So the work in our group takes very complex disorders, schizophrenia, autism, Alzheimer's disease, and we primarily are profiling many levels of gene regulation and genomics across different regions of the brain using a kind of range of novel sequencing approaches. And the real kind of take-home message from this work is that despite the underlying complexity of all these disorders, so no two individuals with autism or schizophrenia probably have exactly the same underlying etiology in terms of their genetic risk or environmental risk exposures. At a molecular level, we're seeing quite strong evidence for a very convergent molecular signature or molecular pathology in the brain. So we've done a lot of work profiling postmortem brain in schizophrenia, and I won't go into too much details on the specifics here. We've had a couple of recent papers published, this work by Joanna Viana. The take-home message is that we see widespread changes at specific sites in DNA methylation. This is data from 4 different brain regions in about 100 schizophrenia brain samples compared to matched controls. But of course, we know that genes don't operate in isolation. In fact, they operate in what we call sort of biological networks. A nice analogy is really to think of the transport network of the northeast US. You can see all these cities interconnected by linear roads on the left, and on the right is a kind of airline hub map. And you could imagine that actually a very major change at one of these positions here, if you think of each of these intersections being a gene, actually probably doesn't have a huge downstream effect on the network. But you could have a very minor effect on one of these kind of core airline hubs, like for instance a big snowstorm in Chicago would have effects across the whole network. And the same is true for thinking about how genes are regulated. So very subtle effects in a very core gene can have very widespread effects on how the system operates. And we see that this structure is really true in the context of genomic data. So we're able to generate modules of co-methylated regions in the genome, in the brain. This is just showing 2 of these modules here that are associated very strongly with schizophrenia in the prefrontal cortex. And what you'll see, firstly, is that these are linked together by a gene called SHANK2. It's probably a gene many of you have heard about. It's a synaptic scaffold gene, very important in brain development and other types of neurobiological function. It's been widely implicated in central nervous system disease. But also these modules are highly enriched for pathways that act on neurodevelopmental processes. The other thing to take home is that the genes in these networks are very highly enriched for the same genes that are being implicated from GWAS studies. And again, this goes back to my point about the power of looking at these molecular signatures, because while these GWAS studies are done on tens, maybe up to hundreds of thousands of individuals, we're able to actually pull out the same gene networks by focusing on much smaller numbers of individuals, but looking actually at the level of gene function in these modular pathways. And the same is true of other neurodevelopmental disorders that we look at. So this is work led by Chloe Wong, looking at autism spectrum disorder. Again, we've profiled DNA methylation across different regions of the brain in cases compared to controls. And you'll see very nicely that the autism cases cluster very, very clearly together. And in fact, this mirrors other work that we've done on these data in collaboration with people like Dan Geschwin, looking at gene expression and other types of epigenetic modification. So again, there's mounting evidence at a kind of molecular signature level, despite the underlying etiology in these individuals, that at a molecular level, that there's some core similarities. And again, in the case of our autism genes, these are very enriched, again, for genes that are playing a role in neurodevelopment. So that really took us on to trying to learn a little bit more about what's going on in normal human brain development and relating that to the diseases that we study. And it's worth noting at this point that there's very limited data actually on what happens in the way that genes are expressed during human brain development. Most of what we know about gene regulation in brain development comes from studies of model systems like mice, but there are kind of key differences between how a mouse brain develops and how a human brain develops. And so we've been undertaking a considerable body of work in collaboration with the Human Developmental Biology Resource and the MRC Brain Bank network that's provided us access to a really unique set of human brain developmental tissues. I think our earliest sample is about 23 days post-conception. Our latest samples are about 180 days post-conception. And we've been profiling all different layers of gene expression and gene regulation in these samples. And again, the take-home message is that this is a period of extreme plasticity in terms of gene regulation. So here are just kind of 4 example sites, and you can see very dramatic shifts in levels of DNA methylation across this period of human brain development. And again, just as I showed before, these changes occur in very coordinated ways. And so we see these are module eigengenes for key neurodevelopmental modules changing across periods of brain development. And these are really dramatic and fast changes. So these are changes that are occurring over the timescale of days and weeks and involve kind of sets of genes that are going from fully methylated to fully unmethylated. And in this case, a module of genes that's going really from fully unmethylated to fully methylated. So these are very dramatic changes. The other thing that we found were that there were very clear sex differences, even on the autosomes, during this period of brain development. So here's just 2 examples of gene regions that differ quite dramatically between males and females across this period of brain development. Perhaps more interestingly are examples like this. This is examples where trajectories of gene regulation across brain development actually differ between males and females. So an example where males and females start off very different at the early stages of gestation and actually kind of converge towards birth, and maybe the opposite example here. And of course, this is interesting given that we know there are clear sex differences in neurodevelopmental phenotypes such as autism. And in fact, this is work that's just been published on bioRxiv last week in collaboration with our good collaborator Nick Bray in Cardiff. And here we have characterized gene expression in exactly the same samples and really find very similar patterns, widespread sex differences in the expression of autosomal genes during brain development, and that these genes influenced by these gender differences are highly enriched for for genes involved in neurodevelopmental phenotypes. So one of the utilities of mapping these patterns of gene regulation across the genome, across development, is that it allows us to start to annotate neurodevelopmentally dynamic domains within genes that we know play a role in neurodevelopmental disorders. So we find that our neurodevelopmentally dynamic loci are very enriched for genes implicated in autism, schizophrenia, and other neurodevelopmental phenotypes, and I've just given 2 examples here. What we're able to see is specific regions within these genes that show very dynamic changes across development, and these allow us to focus our subsequent functional work in the context of these genes on specific domains within the gene. And the other thing we're able to do is to take all the sites in the genome that we find differentially methylated in our adult brains from individuals with schizophrenia. So this is prefrontal cortex data. I apologize, this is quite a complex slide, but essentially each of one of these bars represents a different site in the genome that we find to be altered in individuals with schizophrenia. And on the y-axis is the extent to which that same site is dynamic across neurodevelopment. And the take-home message is that there's a highly significant enrichment for neurodevelopmentally dynamic loci amongst sites in the genome that are differentially methylated in individuals with schizophrenia. Again, this is evidence to really support the notion that there may be neurodevelopmental origins to schizophrenia. And here's just one example here. So this is a site that's differentially methylated in the prefrontal cortex between schizophrenics and controls. And here you can see that DNA methylation at this site is changing very dramatically across this period of brain development. This is in a MYT1L, which is a potent transcription factor that's involved in neurodevelopment. The other types of things that we're looking at in these samples is actually looking at different versions of the gene. So we're very interested in mapping isoform and splicing diversity. So while we may only have maybe 20, 30,000 coding genes, we know that the vast majority of these genes actually show a high level of alternative splicing. This is particularly prevalent in the central nervous system. We know it's developmentally dynamic, and we know it creates proteins that can have very diverse effects, often antagonistic effects in terms of their actual function. Traditional kind of short read sequencing approaches aren't able to actually delineate these different isoform versions, so we're using new 3rd-generation sequencing approaches, things like Pacific Biosciences and Oxford Nanopore sequencing, which gives us reads that enable us to essentially distinguish entire isoforms. And this is one of the most striking results I think I've come across yet. It's kind of doesn't look that impressive here, but this is just a kind of high confidence schizophrenia gene. And these are all new versions of this gene that have never been characterized before and are not seen in common reference data sets. So this tells us that there are thousands of new versions of genes that aren't in existing annotations, and that many neuropsychiatric risk genes are characterized by this extensive isoform diversity in the developing brain. And so I think this is going to be a really interesting area for our future work. So we're not only interested in brain development, we've been looking at gene regulation across the life course. So this is just a kind of summary, really, showing data that we've amassed now on about 1,500 post-mortem brain samples, spanning from those very early stages of development. I think our oldest sample is about 115 years old. And again, this just highlights the very kind of dramatic shifts in gene regulation that take place across the life course, particularly during this prenatal developmental period, but also carry on, but in very different ways in different regions of the adult postnatal brain. And there's 2 things here. One is that there's a kind of big gap missing here. And so this is an area that we're currently concentrating work on, and that's the kind of later stages of pregnancy and the immediate perinatal period. but also that we have a lot of data at the later stages, and that's really allowing us to start to address questions relating to the aging brain and to really delve deep into understanding changes that take place in neurodegenerative disorders like Alzheimer's disease. And in fact, using the approaches that we use to screen DNA methylation across the genome in some of the well-characterized samples that we have access to, we're able to detect really robust and reproducible signatures of pathology for disorders like Alzheimer's disease. This is a couple of papers published a couple of years ago in Nature Neuroscience that really highlighted novel regions of the genome that appear to be differentially methylated specifically in the brain in the context of elevated neuropathological burden in Alzheimer's disease. You can just see here how this is enabling us to really distinguish between cases and controls. And you can also see how reproducible these effects are. So this is just highlighting how the effect size is discovered in our initial discovery sample on the x-axis here, are highly correlated with those in a much larger replication data set from Harvard in the US. You'll see almost clear concordance of effect size. So we're identifying really robust signatures of neuropathology in the brain. The interesting thing is that these changes are all cortex specific, and they're largest in the regions of the brain that are actually affected in the pathology in Alzheimer's disease. And in fact, we don't really see any changes at all in bits of the brain that don't show any neuropathology or in blood samples that we have access to from the same individuals. And the same is true, actually, if we look at other types of gene regulations. This is a paper just published in Nature Neuroscience that's been looking at H3K27 acetylation. This is a marker of enhancer activity across the genome. Basically, elevated H3K27 acetylation is associated with elevated gene expression. And again, you can see evidence that really, if you profile this across the genome, we're able to very clearly distinguish between our Alzheimer's disease cases and controls. And interestingly, from a mechanistic point of view, these changes predominantly map to genes that we know are important in the mechanistic pathology of familial Alzheimer's disease. These are all late onset Alzheimer's disease that don't have mutations in any of these genes. So I think we really are tapping onto mechanisms here. But the other thing is that these were all very specific to cortex-specific enhancer domains. So if you looked in other regions of the brain that weren't affected by the pathology we were looking at, we didn't see these differences. And that brings me on to the next point of this talk, where I just want to kind of highlight some of the issues inherent in doing this type of work, because studies of epigenetics are becoming more and more popular and widely published and talked about a lot. And it's worth, for the students here, realizing some of the limitations inherent in doing this type of work. So if you're doing genetics, life is relatively easy because all you need is a blood sample, because your DNA sequence is the same across all tissues and cells. It doesn't matter when you take that DNA sample because it's not changing across your life. It doesn't matter what a person has done during their life because it's not going to affect their DNA sequence. And the disease that you're studying itself won't actually have any impact on your DNA sequence. And none of that is true in epigenetic epidemiology. And in fact, the field itself is really going through an explosive period. But to date, most of the studies that have been published are based on very small numbers. They've often used inappropriate tissues or cells. They've often used suboptimal study designs, and there's a lot of hype and overinterpretation of the results. So I just want to give you a real-life example. So this is a study of schizophrenia that we've done. This is looking at peripheral blood DNA. We've profiled DNA methylation across the genome. You've probably all seen plots like this before from genetic studies. This is a Manhattan plot. Essentially, each of these dots is a position in the genome. This is significance here, and things above this sort of magic red line are things that you're very interested in. And you can see we found a number of sites in the genome that appear to be very robustly associated with schizophrenia, and you can put names to these, gene names to these sites and start kind of talking about maybe the biology that's going on. And what we noticed was that this Manhattan plot here is actually identical to this Manhattan plot here. And what this is a Manhattan plot from an epigenetic study of smoking. And of course, as you probably all know, epidemiological studies have shown that smoking rates are much higher in schizophrenic patients compared to controls. And so what we've really inadvertently done here is a study of smoking rather than a study of schizophrenia. So the key message is that study designs that are being really used for genetic epidemiology are unlikely to be optimal for molecular epidemiology. So this is true of all sorts of molecular readouts, not just epigenetics. But actually, you can turn that round. So actually, these smoking signals are so robust that we can actually derive an exposure metric from the data itself. And so if I was to take a blood sample from any of you sat here, I would be able to work out if you were, with very high confidence, if you were a smoker or not, or how much you had smoked for, maybe when you gave up smoking. In fact, we can very clearly distinguish between our smokers and non-smokers based on this methylation smoking score. And this is a really powerful thing. So just to show the effects, this has important public health implications. This is data from a study we've just published in Genome Medicine that looked at neonatal birth spots, about 1,500 of these from Denmark, where we were able to map the epigenetic data back to whether the mum smoked or not during pregnancy. And you can see highly significant difference in our methylation smoking score between the offspring of mums who smoked compared to those that didn't. So this is really showing that the maternal environment is having direct effects on gene regulation in the offspring. So another issue that can really confound this type of work is cellular heterogeneity. So I've already told you that different cell types have different profiles of gene expression and gene regulation. And of course, a lump of cortex is made up of many different neural cell types. A blood sample is made up of many different cell types that you can see very clearly. And each of these will have a very different gene expression profile and a different epigenetic profile. And because these levels of cells can also be influenced by things like infection, disease, age, stress, diet, exercise, whatever, even the disease itself, it could be another very important way that you can really get into trouble in confounding your results or interpreting data. So we are trying to tackle this in many ways. So in the context of the brain, where we're particularly interested, we're developing methods for actually purifying cell types from bulk tissue using a range of different antibodies, nuclear antibodies for different cell types. And just to kind of show you, what a dramatic effect this has. So this is essentially a DNA methylation profile from a lump of prefrontal cortex. And then this is from the same individual, the profile from these isolated cell types that comprise this bulk tissue. So you can clearly see if you were studying a disease like Alzheimer's disease, where you may be losing neurons, what you might be doing by running one of these assays is not so much a study of the disease, but actually a very expensive cell count that's really not necessarily telling you much. But again, we can use these data to our advantage and actually derive, using these pure cell populations, derive indicators that effectively allow us to deconvolute the cell proportions from either a blood sample or a brain sample. And we can then use these to estimate cell type proportions from bulk tissue. Just to show that this works, this is looking at neuronal proportions in Alzheimer's disease and controls, and you can see, as you would expect, much lower levels of neurons in our Alzheimer's disease cases compared to controls. And this is from a bulk tissue. So we're really lucky, I think, especially during my time here, and I work very closely with the Brain Bank and other brain banks in the MRC network in the UK, that we have access to very fairly well phenotypes and relatively good quality post-mortem tissue. But of course, if it turns out that we need the numbers for epigenetic studies that have been required for genetic studies, then we're going to be very limited by what's possible using brain tissue. And so we're doing a lot of work to try and work out the extent to which inter-individual variation that's detected in peripheral cells, such as blood, can tell us about variation that's taking place in those tissues. But I still think blood has a potential utility in telling us about biomarkers of disease and telling us about the consequences, maybe not necessarily the actual underlying causes of disease. So we're doing some fairly large-scale studies looking at schizophrenia. So this is a study we've done in collaboration with many people. It's not on the scale of genetic studies, but for an epigenetic study, we have a sample size of about 4,500 cases and controls. That's a fairly large study. Just to kind of, again, highlight what you can derive from this data. So it shows you that across all the cohorts that we've looked at, that schizophrenics smoke more than controls. I guess that kind of highlights the importance of covariating for that in our analysis. And again, we can look at the different cell types. So one interesting thing that's come out is that we see altered cell type proportions in our cases compared to controls, particularly in the levels of granulocytes, which I think is particularly interesting given that we don't see this change in cohorts of samples that are first episode schizophrenic patients that are not on major medication. It's telling us that maybe the medications these individuals are having effects on the blood cell proportions. And again, we can do we can look across the genome. We can find sites in the genome that are differentially methylated in blood compared to controls, and we find a lot of these given the large sample size that we have. This just gives an example of one. This is the top-ranked site. It's annotated to a gene that's actually been previously implicated in autism, and you can see the very consistent effect size across the different cohorts that we've looked at. And this just gives you a kind of list of other top genes that we've found. Again, I won't go into details other than to say that we find a number of genes that have been implicated from genetic studies, and we're currently following those up. And again, these findings are very consistent. So we can take these differences that we find in our initial case control studies, meta-analysis across these 4,000 samples, and show that they're highly correlated in a second independent set of samples. So this is just the correlation of effect sizes. And we also find that these effects are very correlated when we compare them to an ongoing study of discordant MZ twins that we're doing. And I'll say a word about twins because we do a lot of work on twins. Twins are a really powerful tool for doing molecular epidemiology because they allow us to control for many of the things that I've talked about that can really confound the work we do. They share the same genotype, same parents, they're obviously the same age, same sex, and to a certain extent they've had the same early and prenatal environments. And in collaboration with Robin Murray and others, I think we're looking at probably the largest collection of discordant schizophrenia MZ twins. And again, just to highlight that we see fairly robust differences. This is our top-ranked difference annotated to a histone deacetylase gene, HDAC4. That's actually quite interesting because that's a potential drug that's being used for various CNS disorders. And just to show that we see differences, very consistent differences. Each of these bars is an individual twin pair, and you can see very consistent differences of effect across pretty much every discordant MZ twin pair that we see. So twins also allow us to start assessing the extent to which genetic variation influences gene regulation. And so in collaboration with Louise Arsenault and other people involved in the ERISC study, we've been profiling DNA methylation in a large collection of monozygotic twin pairs and comparing the patterns to those that we see in dizygotic twin pairs. And that enables us to apply kind of quantitative genetic analyses to look at the extent to which MZ twin pairs are more similar to each other than DZ twin pairs. And that allows us to identify sites in the genome whereby genotype itself directly affects the levels of DNA methylation. So here's an example here. We call these DNA methylation quantitative trait loci. Each one of these columns is a different genotype group, and you can see that genotype is very strongly associated with DNA methylation. We also find examples like this where actually the correlation is the same in MZ and DZ twins, and these are pointing us towards regions of the genome that are potentially very environmentally sensitive. These would be regions that maybe respond to things like smoking and other types of exposures. And so we can take these molecular QTLs and actually start to use them to understand the mechanisms linking genetic variation to phenotypes. And of course, there are two ways by which a genetic mutation could influence phenotype. It could either have a direct effect on coding proteins, or it can more indirectly and more subtly influence levels of gene expression and gene regulation. And we know for common traits like schizophrenia that these are in the minority. Very few of the GWAS variants for schizophrenia actually have a direct effect on the protein itself. And that most of the changes are actually taking place are more subtly affecting processes like the epigenome and regulating gene expression in that way. And so we can start to put together findings from GWAS together with these maps of gene regulation in relevant cells and tissue types to start generating hypotheses about function. So for instance, if we take regions of the genome that we characterize as being active enhancers, defined by H3K27 acetylation, and then we can look at the extent to which genetic variants associated with a trait are enriched in these regions. This is data from cortex. We see that for all the central nervous system GWASs that we've interrogated, there's an enrichment of heritability within these regions classified as enhancers in the cortex. And what's really interesting is that this is very specific to the brain traits. So if you take other GWAS traits that are non-CNS, you don't see, or largely don't see, such an enrichment. So it's telling us that the genetic variants associated with CNS diseases are likely to be important, enriched in regions of the genome that are regulatorily important for that cell or tissue type. So going back to our fetal samples, we were able to look at the genetic effects on DNA methylation across brain development. So not only characterizing normal patterns of gene regulation, but looking at inter-individual differences. And again, we find very widespread genetic effects on DNA methylation. Most of these occur in cis, so that the genetic variant is very close to the epigenetic change that it's introducing, but a large number of them actually occur in trans. So it's a genetic variant influencing gene regulation on a different chromosome. And that has implications for how we interpret the findings from GWAS, because it tells us that the gene most close to the SNP is not necessarily the gene that's influenced by that genetic variant. And the other thing is that these tend to be very stable across development and across different regions of the brain, but they're not always that way. And so we find examples where a genetic variant can have an effect on gene regulation specifically in the developing brain, but not in any of the adult brain regions that we've looked at. Or conversely, could have actually an opposite effect in the fetal brain compared to a different region of the adult brain. So it's telling us that in many cases, the effects of genetic variants on gene expression could be very developmentally and tissue type dependent. And so we're able to take our fetal brain MQTLs and say, are the genetic variants associated with schizophrenia enriched amongst these genetic variants? And indeed, they are. So it's telling us that the genetic risks for schizophrenia are enriched for genetic effects on gene regulation in the developing human brain. And this is specific to schizophrenia. So if we take a neurodegenerative disease where you wouldn't expect a neurodevelopmental component, or two traits not involving the brain, then you don't see any such enrichment. And in collaboration with Nick Gray, again, this is a paper that was published a couple of weeks ago, we see exactly the same for expression QTLs in the developing brain. And finally, we can use these molecular QTL data to start to actually fine map genetic regions that are associated with diseases. So one of the kind of issues with GWAS, I think, is that you often find these big, large regions of the genome that are associated with disease, and these regions may contain 2, 3, 4, 5, maybe 10 different genes, and actually working out which is the actual gene involved in disease is really the next step. And so we can start using co-localization analyses to find variants that are actually associated both with gene expression or gene regulation and the trait that you're interested in. And so here's just one example gene region where we're able to map this down to a high confidence risk gene within that larger region. And these genes are then good targets for us to do our subsequent functional analyses. And so we're doing things like CRISPR gene editing and showing that these are having dramatic effects both on gene regulation and gene expression and also function of those cell types. So really, I hope I've shown you that traits, common disorders like schizophrenia are more than just genetics in the environment, that we need to look at the mechanism that brings those together. We're particularly interested in the role that epigenetic variation plays in mediating those. And there really is the kind of final point to make. Why are we ultimately interested in this? Well, because actually finding these epigenetic and gene regulatory differences associated with a trait opens up new avenues potentially for developing new therapeutics and new drugs. So we know that the epigenome is much easier to target and manipulate than the actual genetic DNA sequence, and we can design drugs that actually interfere with the actual epigenetic machinery in the cell, either the mechanisms that write the epigenetic marks, the mechanisms that erase those marks, or the mechanisms that actually read those marks. So in combination with Lilly, we're currently screening a whole range of neuropsychiatric drugs in a range of different cell types to actually explore the gene regulation patterns that map onto these different genes, these different drugs. And this will give us ideas about the mechanisms by which those drugs work, but also enable us to design new drugs that maybe can target those same pathways without maybe some of the severe side effects. And just to show you, again, that these drugs really can have dramatic effects on gene regulations. This is a collaboration with James McCabe here as part of the CRESTAR study, which has taken treatment-resistant schizophrenics who are just being prescribed clozapine for the first time and showing very dramatic shifts in gene in DNA methylation as individuals go through their treatment regime. Again, this is highlighting regions of the genome that are being targeted by the drug, and I think that's useful information because the mechanism of action is not necessarily known. But this also tells us that medication use is an important thing to bear in mind in the context of our work. And this is just work on zebrafish brain, again, where we can see gene networks being created and removed by exposure to drugs like clozapine. So this is my final slide. I just want to highlight really how I think epigenetics can have importance in the context of personalized medicine. I hope you've seen that there potential for epigenetic biomarkers of disease. These marks can give us information about exposures. They can perhaps tell us something about mechanisms of disease. They can help us refine traditional risk models. They can maybe tell us something about biomarkers of drug response. And ultimately, the hope is that we can modulate the epigenome via environmental, lifestyle, pharmacological interventions. So again, I just want to put up the slide of my team and acknowledge the people who fund our work. Thank you.
Okay, so one of the world authorities on genetic and environmental factors in schizophrenia is Sir Robin Murray. He's going to say a few words of thanks to Jonathan.
So thanks very much indeed, Jonathan. That was a great talk. You heard from Phil the fantastic track record that Jonathan has got, but you didn't, of course, know that he was a wonderful lecturer. But now you know that, and that he can very readily take very complicated terms that we've never heard of before and translate them into in a user-friendly way so that psychiatrists and sociologists and psychologists who know very little about genetics can understand them. So it's been great to hear this, but Jonathan is not only an expert in molecular biology, but he's also surprised. I don't mean to insult basic scientists, but sometimes you get basic scientists who are clearly fantastic in their field, but they have absolutely no idea about psychiatric disorders. And Jonathan is actually surprisingly knowledgeable. I'm sorry, I don't mean surprisingly for looking at you, but for your type about psychiatric disorders and epidemiology. I mean, how many epigenetic geneticists are designing studies in which they take into account not only psychiatric disorders, but also the effects of smoking and of the different antipsychotic drugs. So this is a sophisticated man who understands not only his own field, but our field. So the other thing, of course, is he's very good with, do you do your own slides? Yes. So the slides are very good and very clear, and explain the relationship between risk genes for schizophrenia and methylation and gene expression. Very nice analogies. I thought showing how methylation is to gene expression is the equivalent of leaves on the line at Clapham Junction, and the differences at different times of life are very interesting, from fetal life through to adolescence. And I was actually wondering, is the railway analogy of adolescence the introduction of new timetables when there's lots of things which may potentially go wrong? So, being my age, I like the historical bits, and I knew both James Shields and Gottesman, and I can't resist telling you one thing about each of them. So when James Shields died, he ran the genetics unit here, which was supported by the MRC in 1979. The MRC had a nationwide review of psychiatric genetics, from which they concluded it was a totally useless subject and should never be given any funds again. Amazingly. So it just shows you that the great and good can make the most stupid mistakes. So fortunately, we were able to keep it going a little bit. The other thing I can tell you about Eric Gottesman was that he did his PhD here in the 1960s. And in 2002, he came back to visit here, and I took him on a tour of the Institute. And as we were going along the east wing, the first floor, we passed a pile of folders and data. And as we passed by, I saw he was looking down at them, and he said, Gottesman said, "That's my handwriting. This is from my PhD. They're not throwing it out, are they? Because somebody had put it in a great pile outside their door." So I said, "Oh, no, they're just moving to a new office." But you would not have believed that somebody coming back nearly 40 years later would be able to spot them. So actually, I guess it shows you should keep very good records. So we've had a very interesting lecture, but it's been really about patience. There's been nothing to learn about how we should choose lecturers or professors or even Janssen lecturers. So are we moving into a dystopian future where instead of just looking at how many GCSEs lecturers got, did they have a first class or a 2-1? Should we just ask for a little sample of blood? And we'll be able to run apologenic risk score, and if you are high on an apologenic risk score for IQ and low on apologenic risk score for psychopathy, you get the job. But what about professors who have gone through more of their life? I think epigenetics would be more important. If you're choosing a professor, you want to know how, or certainly a young professor, you want to know is he smoking? And it's of course no use asking because he or she, you want to know whether they've been smokers. So you would just do epigenetics, and you would find out the smoking signature, and you'd have an idea how long they might last as a professor. But similarly, you might what about the alcohol signature? Would you want professors who had who evidently had never drunk in their life, or would you like heavy drinkers? Well, I'm reminded that our lecturer this evening often, at least the meetings of the collaborators that I've been at, have usually occurred in pubs. But so you're obviously quite fond of pubs, but while you've been there, I've noticed you haven't drunk very heavily. So I think you would just have a modest effect on your epigenetic signature for alcohol. So anyway, it's been a great tour de force, and those of us who have struggled with epigenetics and methylation and gene expression, I think I won't say I won't struggle at all, but I'll be ignorant at a much more sophisticated level than I was previously. So thank you very much indeed, John.
Thanks very much, Robin. So thanks again for everyone for coming. I want to just thank Janssen for generously sponsoring this meeting, as they always have done, and let you know that there's a reception upstairs afterwards, which you're very welcome to attend. Thanks very much for coming.